Today we will introduce DNA replication and protein synthesis. First is DNA replication. Remember, DNA replication must occur before a cell can divide. Next, we will introduce the enzymes that will be used in this process: helicase, primase, and DNA polymerase, exonuclease, and ligase. This is DNA, and this is what it will look like when it is unwound. First, helicase will separate DNA into two strands by breaking the hydrogen bonds. Here, the top strand is called the leading strand and the bottom strand is called the lagging strand. These two strands run in opposite directions. Primase attaches free nucleotides in a short strand of RNA called a primer. Then, DNA polymerase bonds to the primer. It adds DNA bases to the strand working from the 5' end to the 3' end. It adds complementary bases, meaning thymine is added to adenine and cytosine is added to guanine. In the leading strand, this is a simple and uninterrupted process. The lagging strand has to go through a more complicated process. This is because it runs in the opposite direction, which would require DNA polymerase to build from the 3' end to the 5' end. It can't do this, so primase returns, making several primers. Then DNA polymerase works from primer to primer. The segments it builds are called Okazaki fragments. Exonuclease comes and removes the primers, so DNA polymerase can come and fill in the gaps. Then ligase bonds these segments. There is always one side from the original strand of DNA and one that is new. This is called semi-conservative and is to limit mistakes by using a template. Protein synthesis. This process begins in the nucleus. Within the strand of DNA, there are segments called genes, which hold the instructions for the making of proteins. DNA is separated, and RNA polymerase adds the complementary base temporarily. This strand is called messenger RNA. The mRNA travels from the nucleus to the ribosome and the cytoplasm. Once in the ribosome, the mRNA is read in a 3-base segment called a codon. Based on the codon, the correct amino acid is brought in by the tRNA. The amino acids together form a polypeptide chain. The chain begins to fold into spirals, which then folds into a complex 3D shape to form a protein. Sometimes this protein has more than one polypeptide chain.